and School of Pharmacy established in 2010. Due to the various study programs, medical faculty has been renamed into Faculty of Medicine and Health Sciences. Faculty of Medicine and Health Sciences at University Muhammadiyah Jakarta.
Our vision is to be Faculty of Medicine and Health Sciences with good faculty governance and independence, evidence-based in developing science and technology, root from Islamic Indonesian social culture, beneficial for people and including one of the best in Asia.
Interprofessional education is one of top program in Faculty of Medicine and Health Sciences Universitas Muhammadiyah Yogyakarta. The existence of 4 health professions in Faculty of Medicine and Health Sciences Universitas Muhammadiyah Yogyakarta is a valuable opportunity to organize collaborative practice during courses either through undergraduate course or through profession phase. The existing Center of Islamic Medicine Study for Faculty of Medicine and Health Sciences Universitas Muhammadiyah Yogyakarta is an effort to realize integration of Islamic value into medicine and health.
The relationship of Faculty of Medicine and Health Science Universitas Muhammadiyah Yogyakarta consists of several affiliations with various institutions within country and abroad, either public or private.
Not only to learn, but also to understand. Therefore, there are time and space for us to comprehend health as science.
What I like the most is that we have passion in health. It is about the quality of the education itself. To love health as an occupation will make us ready
Assalamualaikum. Tes. Assalamualaikum.
Waalaikumsalam.
Kedengaran ya?
Iya, kedengeran.
Devi nih atau siapa?
Iya, Devi.
Oke. Tak coba pakai headset juga ya.
Oke.
Bentar. Suaranya Hamdan kedengaran nggak, Dev?
Enggak, tapi suara Bu Anissa jauh.
Oh, oke. Kalau gini?
Udah. Cukup.
Enggak.
Sudah cukup. Enggak, udah jelas.
Oke, sebentar ya.
Profnya masuk.
Medicine and Health Sciences at University Muhammadiyah Yogyakarta towards the world of health services in Indonesia. Several kinds of achievements obtained by students and lecturers of Faculty of Medicine and Health Sciences in field of health. Faculty of Medicine and Health Sciences at University Muhammadiyah Yogyakarta will continue these achievements in order to support and improve quality of Islamic medical education and health study in Indonesia.
Join us to reach the excellence and international competitiveness in medicine and Islamic health.
Welcome to Yogyakarta, a city with culture and local wisdom. The culture and local wisdom offered in Yogyakarta become the main factor for students to visit and to pursue their education in this city. In March 1993, Central Board of Muhammadiyah has established Medical Faculty of University Muhammadiyah Yogyakarta to educate potential medical doctors. Along with the demand on other health personnel in the society, School of Nursing has been established in 2000. School of Dentistry has been established in 2004. And School of Pharmacy established in 2010. Due to the various study programs, medical faculty has been renamed into Faculty of Medicine and Health Sciences. Faculty of Medicine and Health Science
Our vision is to be Faculty of Medicine and Health Sciences with good faculty governance and independence, evidence-based in developing science and technology, root from Islamic Indonesian social culture, beneficial for people and including one of the best in Asia.
Interprofessional education is one of top programs in Faculty of Medicine and Health Sciences, Universitas Muhammadiyah Yogyakarta. The existence of 4 health professions in Faculty of Medicine and Health Sciences, Universitas Muhammadiyah.
Assalamualaikum, Mbak Dian, Mbak Nisa. Kula pun gabung. Kok orang nang salju, Mbak? Oh, saya suaranya ra metu. Tak kira nang burine ana saljune, Mbak Nisa.
Ya Bu, ini putih-putih. Sudah bisa kita mulai apa belum ya videonya tadi? Halo.
Jakarta consists of several affiliations with various institutions within country and abroad, either public or private institutions, in order for the development on academic, research and community development. It is an evidence of trust from public toward Faculty of Medicine and Health Science, Universitas Muhammadiyah Yogyakarta. This relationship is conducted to support the faculty in educating and establishing young health professionals worldwide. The students' activities in Faculty of Medicine and Health Sciences, Universitas Muhammadiyah Yogyakarta are designed to establish health professionals who are able to compete globally without losing Islamic value. Thousands of scholars from Faculty of Medicine and Health Sciences, Universitas Muhammadiyah Yogyakarta are academically tested and work in various and dynamic activities. This makes the alumni strong and competent.
Faculty of Medicine and Health Sciences, Universitas Muhammadiyah Yogyakarta implements the method that we are educated not only to learn but also to understand. Therefore, there are time and space for us to comprehend health as science.
What I like the most is that we have passion in health. It is about the quality of the education itself. To love health as an occupation will make us ready and professional in working world later.
This faculty, I find what Indonesia is. I mingle, I share, I interact with students and lecturers. I find it really nice and full of tolerance. I think the educational system is not different with my country.
It is a living contribution from Faculty of Medicine and Health Sciences at University Muhammadiyah Yogyakarta towards the world of health services in Indonesia. Several kinds of achievements obtained by students and lecturers of Faculty of Medicine and Health Sciences in field of health. Faculty of Medicine and Health Sciences at University Muhammadiyah Yogyakarta will continue these achievements in order to support and improve quality of Islamic medical education and health study in Indonesia.
Join us to reach the excellence and international competitiveness in medicine and Islamic health.
Welcome to Yogyakarta, a city with culture and local wisdom. The culture and local wisdom offered in Yogyakarta become the main factor for students to visit and to pursue their education in this city. In March 1993, Central Board of Muhammadiyah established Medical Faculty of University Muhammadiyah Yogyakarta to educate potential medical doctors. Along with the demand on other health personnel in the society, School of Nursing has been established in 2000. School of Dentistry has been established in 2004. And School of Pharmacy established in 2010. Due to the various study programs, medical faculty has been renamed into Faculty of Medicine and Health Sciences. Faculty of Medicine and Health Sciences at University Muhammadiyah Yogyakarta commits to providing graduates as outputs who are professional, Islamic and able to develop science and technology on medical and health fields for the welfare of the nation and Muslims, as well as able to compete worldwide.
Okay, good morning, Professor Okino, Professor Tatsufumi Okino.
Good morning.
Good morning, Sensei.
Good morning,
How
everyone.
are you, Sensei?
I'm pretty good. I'm very good. How are you?
I'm fine, thank you, Sensei. And maybe we can start for the public lecture today. Maybe, is it okay, Miss Devi? Can we start the lecture today?
Yes.
Yes, okay. Yeah. Okay. Thank you very much, Sensei, for joining with us. And today is still we have atmosphere of New Year, so we would like to say Happy New Year, Sensei. Akemashite omedetou gozaimasu.
おめでとうございます。
Omedetou gozaimasu. Okay, let's start the public today. Assalamualaikum warahmatullahi wabarakatuh. Good morning, Professor Tatsufumi Okino. Good morning, Head of School of Pharmacy, Pharmacist Saptanti Harimurti, PhD. Good morning, Head of Pharmacist, Profession Education, Pharmacist Nurul Maziah, Master of Science. Good morning, lecturers and all of the students of School of Pharmacy, Faculty of Medicine and Health Science, Universitas Muhammadiyah Yogyakarta. I hope all of you are doing good and in a good condition and healthy. We are happy today because we will have a special guest lecture from Hokkaido University, Japan. Thank you very much for the coordinator of this event, Pharmacist Pinasti Utami, Master of Science, for preparing this public lecture. And here I will facilitate or moderate this class, and we hope that we can learn many things from this lecture. Okay, so the topic of the lecture today is the opportunity of the marine natural products in the pharmacy or pharmaceutical science. And we know that the earth provides so many natural products such as marine products. And we can explore natural products to find some materials that give benefit for the human. So how is the opportunity in the pharmaceutical field? And this is very interesting topic since Indonesia has a high biodiversity in nature, also in marine life. So it is challenging to have them to be explored. This topic will be delivered by our special guest lecturer, Professor Tatsufumi Okino. He is a professor from Graduate School of Environmental Science, Hokkaido University, Japan. And Professor Okino is an expert in marine natural product chemistry. So many research have been done by Professor Okino, and especially the biologically active compound from marine life. Before we start the public lecture, we will listen the speech from the head of School of Pharmacy, Pharmacist Saptanti Harimurti, PhD. The time is yours.
All right, thank you very much. Good morning, Mr. Professor Tatsufumi Okino. Konnichiwa, Prof.
Good morning.
Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. It is a very great pleasure to me to welcome you to this general lecture, and we are very thank you that you are taking time out for your very busy schedule, I think, to join in this class today. And for your information, Prof, that there are around 400 students joining in this class, maybe in the Zoom platform is around 100 and another is in the YouTube. So, so many students are ready to join in this class. Hopefully, after the end of the class, all the students will have a wider understanding on the opportunity on what to say, the developing of new drug from the marine products. Yeah, as we know that Indonesia is very rich with the marine biodiversity. So hopefully after this, all the students will more understand about that. So last, I would like to say to thank you to the committee to organize this guest lecture. And one more, Professor Tatsufuni Okino, thank you very much. Yoroshiku onegaishimasu. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Pharmacist Saptanti Harimurti, for the speech. And now we will have the lecture from Professor Okino, and we will have a discussion session for 15 or 20 minutes after the lecture. And for the audience, please prepare your questions. Professor Okino will be very happy to answer your questions. Okay, I think Sensei can start to show the presentation, or from Devi.
Oh, can I start now?
Yes.
Okay.
Okay. Maybe you can try to show the PowerPoint first.
Okay.
Thank you.
Yeah, thank you so much. Thank you for I'm very happy to give a lecture to Universitas Muhammadiyah Yogyakarta. So it's my great honor to be here. So I visit Yogyakarta maybe every 2 years in the past 10 years. So this is the first time to visit Universitas Muhammadiyah Yogyakarta through Zoom. So, well, so today's lecture, I will explain the history or current status and future of marine natural product development in pharmaceutical science. So let me share the PowerPoint. I think you can see my PowerPoint. So again, I'm Tatsufumi Okino from Faculty of Environmental Earth Science and Graduate School of Environment Science of Hokkaido University in Japan. So let me introduce myself first, or let me introduce Hokkaido University first. So I don't have a video for introduction of Hokkaido University. However, but just in case, I think you are here somewhere, then probably you know where Japan is. Then our university is, the name of our university is Hokkaido University. So Hokkaido is the name of this island, okay? Then the biggest city of Hokkaido is Sapporo. So Hokkaido University is located in Sapporo. So maybe we are struggling to prepare the Summer Olympics in Tokyo, but I think, but also we are planning to have the Winter Olympics in Sapporo again, maybe in 2000, I don't know, 30 or 34. So, okay. I cannot move the point. Please wait. Okay. So this is the history of Hokkaido University. So, well, Hokkaido University is one of the national university, or yeah. So then it was established in 1876 as Sapporo Agricultural College. Actually, that time, the Hokkaido, only a few people lived in the Hokkaido. So then we started to develop the Hokkaido during this time. So many of people came to Hokkaido from the United States to establish cities and towns, or to start agriculture and so on. So that's why the vice president of Sapporo Agricultural College is Dr. William Clark. So he is very famous person in as a for Hokkaido University. So then, yep. So let's see. Actually, this is a kind of monument, Sapporo Clock Tower. Right now, it's not located in the campus, but and it's just right now it's a monument or the small museum. But this is very famous clock tower in Sapporo, and actually it was a part of Sapporo Agricultural College. So if you have a chance to go for a trip to Sapporo, maybe you might visit our campus and this clock tower. So anyway, this is the campus. This is a botanical garden. Then actually, maybe from here to the bottom is the downtown of Sapporo. Then it's very close to the downtown, and then it's very wide. And then our graduate school is located here. So we have, I think, 18,000 students and 4,000 staffs. Okay. So, again, this is a graduate school of environment science. So it was established in 1977. I think the first environmental science oriented graduate school in Japan. Okay. Then actually we renamed again. Then actually then we went back to the original name in original English name, but Japanese name of original Japanese name is a little bit different from the current name, but English name is the same now. Okay, so we have around 500 graduate students, only graduate students. So we have 4 divisions. So from us, division of environment science development is kind of integrated science. So also we have a division of earth system science, biosphere science, environmental material science. So most of natural science is covered in terms of the environmental science. Okay. So this is a photograph of Hokkaido University campus. So this is autumn. It's very, very beautiful. Maybe this is early summer. Again, it's very beautiful. In terms of the environmental science. So this is a photograph. Okay. So is the voice is okay? my voice is okay.
Okay, Sensei, that's fine. Okay, Prof.
Okay. So this is winter. Currently is covered. I'm hearing my voice, so I'm okay. So the town and the campus is covered with snow, so it's very cold. But yeah, if the snow when the snow falls in the evening, it's very beautiful. Okay. So then let me introduce myself. So my research interest, actually I'm a natural products chemist. So basically I'm looking for new compounds which can be useful compounds, including the medicines, right? So then especially chemistry of cyanobacteria or blue-green algae is my the largest part of my research. Okay. Also, recently I'm doing a biosynthesis of halogenated compounds from the red alga Lawrencea. Actually, marine natural products have many unique characteristics. Then one of them is the number of halogenated compounds. So we can find many halogenated compounds from marine sources. Of course, the seawater contains lots of sodium chloride, so you can imagine you can isolate many of chlorinated compounds. But in fact, we can find many brominated compounds. So right now, I'm doing the research. I'm trying to characterize the halogenates from the algae. Okay. So, actually, I will talk these topics later, but this is a very good example. So this compound is now in the market as an anti-cancer agent, but as a antibody drug conjugate. Okay, then this compound was developed from the cyanobacterial compound drastatin 10. Okay, so then this compound shows the very good example to this compound is a very good example to show the potential of cyanobacterial compounds. Anyway, I will show this later. So that's why I'm really interested in the cyanobacterial secondary metabolites. So I'm especially the cytotoxins or enzyme inhibitors rich in cyanobacteria. So that's why we are screening for the cytotoxins and enzyme inhibitors mainly. Okay. Then, but anyway, probably most of students in this class will use drug, right? Or will prescribe the drugs. So, but my interest is looking for drug candidate from natural source, especially marine sources. So maybe you might learn how to find the drug candidates from natural sources. So that's the point of this class. Okay, so where are we now? Let me check. Okay, so well, this is kind of very famous graph, but I copied from the Journal of Natural Products. this is a very updated version of the number of approved drugs in the last, I think 40 years, right? Then, but each color shows the source. Okay, so for example, this P is biological macromolecule, but N, NP, ND, they are natural products based compounds. So N is unaltered natural products, so purely natural products. NB is botanical drug. So maybe we can say the mixture of natural products. And ND is natural product derivatives. So maybe it's difficult to the natural product itself as it is for the medicines. However, derivatives of natural products is used for medicines, right? So even this is a synthetic drug, but this one is synthetic compounds, but a mimic of natural products. Also, this is also a synthetic drug with natural product pharmacophore and mimic of natural. So it means, so this is, we can say natural product, right? But also this part and this part. So let's say 1/3 of approved drugs is related to natural product. Okay, so purely synthetic compound is only 25%. Okay, that's the reality. Also, this is the history of all new approved drugs derived from natural sources. So in 80s, yeah, many of compounds came from natural sources, but even recently, this one, blue one, light blue is ND, so derived from a natural product derivatives, right? Then usually it's a semi-synthetic modification. So semi-synthetic modification means natural. Basically, they use a natural products, but they change the structure. So they modify the natural products. So they use the natural product. Okay. So then, yeah, even recently, this is 2019. So even recently, many of new drugs are from natural sources. Okay. So many people might misunderstand most of drugs comes from the synthetic or synthetic compound, but reality is many of drugs are derived from natural sources. Okay. This is Marindit is a famous database of marine natural products from right now under the Royal Society of Chemistry. Unfortunately, excuse me, unfortunately, this is not a free database. So without the paying, maybe by library, you cannot use it, but it's a, this database contains only marine natural products. Okay. But actually, annually, they publish the updated review in natural products reports. So if you are interested current status of marine natural products research, it's good to read the review published in natural products report every year. Okay, so actually, I checked the marine lit on the 1st of January this year. Then Actually, currently, the total number of reported marine natural products is 36,000. Okay. When I was a student, the number was 3,000. Then, wow, 3,000, so many compounds are reported. That's a common word for the introduction of marine natural products articles, but now it's 10 times more. Okay. So then, 32% of marine natural products is derived from porifera, so mainly sponges, okay, or 16% derived from Cnidaria. So Cnidaria contains a lot of animals, but mainly corals. Okay. So how about my fun cyanobacteria is 3%. Okay. So it's small, but still, you can find it's not included in others. Some significant contribution. Okay. So this data shows again the number of publications by year, right, and by sources. Okay. So then maybe it started around 1960s. So marine natural products research started in 1960s. So actually, I think maybe before penicillin's discoveries, natural products research only focused on the plants. Okay, then after penicillin was discovered, I think 1950s was the era of antibiotics. Then 1960s, natural products research started. Then probably 1980s or 90s was the boom of marine natural products. Okay. Then again, porifera sponges is the most abundant, but yeah, interestingly, Cnidaria corals publication is still steadily increasing. But Ascomycota microorganisms or actinobacteria, actinomycete, is really increasing in after 2000. Okay. How about cyanobacteria? Yeah, it's increasing. Of course, the percentage is small, but it's still increasing. Okay, and I think rhodophyte, lethargy. I think in the beginning, rhodophyte so is the major source of marine natural products. Then the publication is steady or not increasing. So right now it's small, but I think in the beginning it was easy to collect the lethargy. But after the scuba diving became popular, we can dive. So marine natural products chemists can collect marine animals uh, by ourselves. So then, the number of publications from coral or sponges increased. Okay. How about this data? This data, maybe I don't have a title. Oh, yeah. Actually, it's a simplified version. So just a sesquidecade. So 15 years spans data. Okay. Then again, this is divided by the source of marine natural products. Then, okay. So let's see what is this. Ascomycota mycota is rapidly growing, right? Also, actinomycete is rapidly growing, right? But still, sponges is the richest and coral is increasing. So you can see the same trend more easily. How about this one? This one, actually, I think United States contribute to the marine natural products chemistry significantly. They are the major players. But East Asia, like Japan, China, South Korea, Taiwan, we are also major players in marine natural product chemistry. Especially Japan was so in the 70s or early 80s, we published a lot, especially from let's say corals or sponges or lead algae. So look at this. In this era, lead algae is the most abundant source of natural products from Japan. Okay. Then on that time, China, Taiwan, Korea, almost nothing, right? Except very few publications from corals, right? Then from late 80s to 90s, yeah, sponge is the richest and coral is was followed. And then China, Korea started their research from sponge and corals. Then after 2000, okay, still Japanese scientists are interested in sponges, but the other publication decreased. But China, Korea, they started microorganism research like, actually this is coral, and this is ascomycete or actinomycete. So now, recently Chinese or Korean scientists are, rapidly growing. Then they are interested in marine microorganisms. And interestingly, coral also is important, the importance, so their source. Okay. For me, Nigeria, coral produce a lot of terpenoids. Of course, terpenoids is interesting chemistry, but for me, the structure are similar. So maybe it's difficult to attract attention to our publication from corals. That's why I prefer the cyanobacteria, but for some reason, Chinese people or Korean people are interested in corals. And microorganisms is very understandable because we say that it was difficult to culture the marine microorganisms. That's why we are not interested in this, 20th century. But let's say now it's easier to culture the microorganisms. Also, you can get the genome of microorganisms very easily, right? If you have equipment, you can analyze by yourself, or if you don't, just you can pay maybe like $1,000. You can get the data of genome of microorganisms, right, from the company. So let's say then you can do the biosynthetic research very easily. So that's why microorganisms is very good target in the past 20 years. Okay. So let me start to introduce the examples of medicines from marine sources. So this is RA or RC is the first examples of marine medicines, right? Oh, let me. Actually, I will, I will show the approval year maybe in the following slides too. But, sometimes, one drug is approved in United States, in EU, in Japan, in Indonesia, or in the other countries, right? So it's kind of approved year is, there are many approved years for one drug. So, but I cited the information from the marine pharmacology website. So I will introduce it later. So that's why probably most of the data is the approval in the United States. Okay. So anyway, RC. The history of RC is started from 1955. So I think this is the sponge, Cryptothesia crypta. So scientists discovered these unique nucleoside. So nucleoside is a component of RNA, right? But the structure is a little bit different from the nucleosides of RNA. So they called it spongothymidine or spongogliidine. So I think these sponges was collected in Caribbean seas. Okay, so then by by modifying these chemicals, or they tested modified chemicals, then finally RC or cytarabine. So as you know, the drugs have many different names, trade name or common name and register name. So maybe I will show you multiple names for one drug. So for example, this is RC as well as cytarabine. Okay. So this compound was approved as anti-cancer agent. Okay. Then the biochemical target is DNA polymerase, so inhibition. So by inhibiting the DNA polymerase, RC can inhibit the DNA synthesis. So that's why it can show the cytotoxicity. Right? So RA, or another name is vidarabine, okay, was approved as antiviral in 1976. Okay. Again, this compound inhibit viral DNA polymerase. Okay, viral DNA polymerase. So actually, I think this is the only antiviral drug from marine sources. But right now, antiviral drug is very important, right? So of course, probably the Japanese Prime Minister will declare the state of emergency maybe tomorrow for the COVID-19. So probably Indonesia is also suffering from this situation. Let's say, but the problem, of course, the vaccines have been developed, and then some countries started the injection of vaccine for COVID-19, but also we need effective drugs to treat patients, right? So we can use some drugs, but I think we need more effective drugs. But maybe, as you know, the development of drugs takes a very long time. So as for the COVID-19, we should use the existing drugs. We should choose the drugs from existing ones. So, but I'm sure after the COVID-19, maybe the other viruses will appear. Of course, it's different from SARS or MERS or COVID-19, but probably it's similar too. So if we have a list of or deposit of antiviral drugs, we can fight new emerging, viruses. So that's why not only pharmaceutical industry, also academia should focus on the development of antiviral drugs. So I don't have a citation here, but recently Bill Garwick in University of California, San Diego, UC San Diego, read a review about the natural products which can inhibit the COVID-19 or related viruses. So there are lots of examples. So if you are interested to doing the research of antiviral compounds from natural sources, it might be a good start. Reading this review is a very good start. So the review is published in Journal of Natural Products recently. So anyway, so the marine natural products, this is, the very good examples of marine natural products drugs. So then I think I maybe made mistakes. So here, so this is consonants. Probably, I think the most of the research came from Philippines, but probably in Indonesia, you can find these beautiful mollusks. However, this is very dangerous, very, very toxic, right? So they are slow moving, but they can inject venoms to the fish to eat them, right? So then there are many, many toxins, many different toxins. So we call it conotoxins, right? The conotoxins are peptides, very different, many of peptides. So one of the peptide, this peptide, okay, this peptide can act to the N-type calcium channels, okay? Then different conotoxin target the different channels, calcium channel, or sodium channel, or potassium channel. The target, or there are a large variety of peptide structure and target. Can you hear me?
Yes, we can hear your voice, Sensei.
Okay. It's a little bit strange. But anyway, this compound, this peptide, as a pain medication for cancer patients, okay? Because this compound is 100 to 1,000 times more potent than morphine, okay? Then morphine is very addictive, right? But this compound is not, it doesn't show any addiction, okay? Then, however, peptides is peptide, so it's digestible. So it needs to use a spine injection. So that's why it's not easy to use, okay? But anyway, at least the second example in the United States. Okay, so I think in Europe or in Japan, maybe there are some different compounds are approved before them. Okay, so, and also interestingly, you might know omega-3 acid ethyl esters. Okay, so this is a component of fish oils, especially mixture of eicosapentaenoic acid and docosahexaenoic acid ethyl esters were approved in 2004 as an anti-lipemic agent. Their targets are triglyceride synthesizing enzymes. So if you have a high value of triglycerides, you can use these esters. Of course, right now, many of pharmaceutical companies still doing clinical trials of omega-3 acid ethyl esters. Okay, but of course, I think not only as medicinal drugs, but also you can buy it for the health food or functional food, right? So this comes from a fish oil, but to manufacture these esters as medicines, purity is very important. So that's why the purification of EPA or DHA from fish oil is very important, or actually needs a high technology. Okay. Also, probably not only from fish oil, microalgae like phytoplankton or diatom are very good source of omega-3 acid ethyl esters. So maybe some companies extract these oils from microalgae. Okay. Then more, for me as an organic chemist, I'm more interested in the other compounds. So this is another good example. Halichondrin B. Professor Uemura in Japan reported the structure of Halichondrin B isolated from the sponge Halichondria okadai. So actually you can collect this sponge at the coast very easily. But they extracted 600 kilograms of sponges to isolate to do a structure elucidation of Halichondrin B. Okay. Then right now in 2010, halaven, this compound was approved as anti-cancer agent. So this is very different, but can you realize that this part of Halichondrin B is the same as halaven? Okay, so actually this compound was produced by the total synthesis, so organic chemically synthesized by 62 steps synthesis. It's a very long step, probably the longest step of synthesis for medicines. Okay. Then actually Professor Kishi, again Japanese chemist, but working at Harvard University, collaborated with again Japanese pharmaceutical companies, Eisai or Eisai USA. Then first Dr. Kishi synthesized this compound. Okay. Then after that, of course they did longer synthesis than 62 steps. But anyway, synthesis of this compound is very, very costly, very expensive. The pharmaceutical companies were not interested in this compound. Actually, I said Dr. Uemura, Nagoya University, determined the structure, but originally, Fujisawa Pharmaceutical Companies, a scientist in Fujisawa Pharmaceutical Companies, isolated this compound. Then they know the activity is very potent and very promising as anti-cancer agent. However, preliminary analysis of the structure taught them that they cannot develop it. The structure is too complex to develop as a drug. Okay? Then that scientist contacted to the other chemists to determine the structure. Okay? Then academic researchers determined the structure. Then Dr. Kishi synthesized it, but still no company was interested in. But actually only Eisai was interested in. Then Dr. Kishi provided intermediate of the synthesis. So they synthesized these compounds from small molecules, right? Then they expanded, they enlarged the structure, right? So then after that, maybe I think this right half of the compound showed almost the same activity as helicondrin B. Then as I decided to develop the only half structure for medicinal use. But still, 60-step synthesis is not common in pharmaceutical industry, but they achieved it and then they can sell it now. Okay. I want to show you more history of halaben. So again, as I explained, it was developed by Professor Kishi's group and Eisai, approved in 2010 and marketed by Eisai. And actually the target is metastatic breast cancer. Okay. Then biochemical target is microtubules, which is similar to paclitaxel or Taxol or vincristine, which are common anti-cancer agents, but different mechanisms. Okay. So let's go back to the Drastatin 10. So as in the introduction, I showed the example of secondary cyanobacterial metabolites, Drastatin 10. Okay. So let's see. Okay. So the halaben is a very good example to show the power of organic chemistry. Okay. So then Drastatin 10 is a good example to show the ADC, where is it? Antibody drug conjugate. I will explain it later. Okay. But anyway, let me explain the history of Drastatin 10. It was discovered in maybe 60s, and then the structure was reported in 1972. Originally, this compound was isolated from the sea hare Dolabella auricularia. I'll show the photo later. Then many people question that what is the true producer of this compound. Maybe sea hare doesn't synthesize this compound. Then 2010, this compound was isolated from the cyanobacterium of Symploca species. This is all name. Currently, the name was changed. Cyanobacterium Caldora penicillata. Okay. So then, actually, see here, Durabella auricularia eat cyanobacteria. Then accumulate these compounds in their body. Okay. So in marine natural product chemistry, you can find such a food chain relationships. So if you extract very good compounds from marine animals, in many cases, these compounds are produced by that animals, but the other animals. So this is a very good example. Okay. Then anyway, from the discovery of these compounds, this was very important. So National Cancer Institute, United States, in the United States, invested a lot to these compounds to develop as anti-cancer agent. So in 70s, in 80s, they did some clinical trials, but they failed due to the potent toxicity. Then they changed the structure, they modified structure. So the final candidate is monomethyl auristatin E, or abbreviated by MMAE, right? This compound. But still, this compound itself cannot be a drug, anti-cancer drug, due to the toxicity. So then, let me, the Seattle Genetics, use the antibody drug conjugate. So Adocetris, or other name is brentuximab vedotin. So Adocetris consists of monoclonal antibody which targets CD30 and cathepsin cleavable linkers, spacers, and MMAE. Okay, so these components are conjugated to make this drug. Okay, then actually let me explain this one here. So this is ADC, antibody drug conjugate. So then monoclonal antibody which target to CD30. So antigen is CD30 in the especially expressed in the cancer cells. Okay, so monoclonal antibody carry this ADC into antigen, right? Then then after they go into the cells, in the lysosome, the lysosomal cathepsin cleave this compound. Okay, then from the conjugate, this one is MMAE. Okay, so MMAE is released in the cancer cells. Then as a result, cancer cells die. Okay, so ADC, antibody drug conjugate, is a hybrid drug of monoclonal antibody with the targeting capability and cytotoxic drug with cancer killing ability. So targeted capabilities, so probably antibody target the cancer cell specific antigen. Okay. Then also there is some mechanisms of cleavage of cytotoxic cells drugs, which is the catepsin cleavable linkers. Okay. So by using this technology, I know many of natural products was abundant during the clinical trials due to the toxicity. Okay? But if you can use this technology, maybe you can develop abundant natural products as medicines. Okay, so kind of this is a dream technology of natural product chemists. Okay, so it's developed by Seattle Genetics. So Seattle Genetics is an expert company of antibody drug conjugate. Okay, so then I want to explain the different issues by using this compound. Okay, so this is Dravera auricularia. Okay, I think this was this. But anyway, Drastatin 10. To determine the structure of Drastatin 10, 30 milligram of Drastatin 10 was isolated from 2 ton, so 2,000 kilogram of this sea hair. Okay, so it's really very significant amount. Okay, then this is a photograph of cyanobacterium, the true producer. Okay, so we can culture them. Okay, if we can use the cyanobacteria, that's fine, but 2 tons, collection of 2 tons of sea hair destroys the marine environment. Okay. So also in the case of Helicondrin B, Dr. Uemura collected 600 kilograms of the sponges. Okay. It was okay that time, but now it is not allowed. So maybe we can extract 1 kilogram of sponges, it's okay, but 100 to 1,000 kilogram of animals is not allowed recently. So actually I copied this statement from the author guideline of Journal of Natural Products. Okay, so authors should be aware of the fact that large-scale collection of marine or terrestrial organisms may have negative ecological effects. Okay. So therefore, authors describing an investigation derived from large-scale collection should thus include a statement in the manuscript explaining why the collection had no significant adverse or negative ecological effect, or justifying such effect in terms of the benefit from the resulting work. Okay? But, this is really difficult. Well, in this case, 72, of course they know it's really potent, but they didn't know they can develop these compounds as anti-cancer drugs, the marketed drugs, right? So from the current point, we can say 2 tons of extraction of Drastatin 10 was, understandable, or extraction of 600 kilogram of Helicondria okadai is okay, right? Because they produced the marketed anti-cancer drugs and they saved lots of lives. But when they reported the structure, they didn't know that future, right? So that's why, justifying such effect in terms of the benefit from the resulting work is really difficult. It means large-scale collection is very, very difficult. recently. So then again, the author guidelines said when organisms are collected from a foreign country, the corresponding author must state in the cover letter with the submitted manuscript that formal collection permission was obtained. Of course, formal collection permission is very important. I know the Indonesia government is, strong policy to protect the natural sources. That's why it's, without formal, we don't go to the Indonesia, or actually no scientists go to the Indonesia recently without formal collection permission, right? So related to this issue, I want to explain the Nagoya Protocol on access and benefit sharing. So sometimes we call it ABS. So ABS aims the implementation of one of the three objectives of the Convention on Biological Diversity: the fair and equitable sharing of benefits arising out of the utilization of genetic resources, thereby contributing to the conservation and sustainable use of biodiversity. So, so we need to, so especially, probably many scientists want to collect the, resources or marine animals, marine bacteria from maybe Indonesia, right? But we need to have a prior informed consent, or we need to have a contract including benefit sharing. Okay, so after making this, consent and contract, now we can go to that country to collect the organisms. Then after we return to our country, we need to report to our government. So we collected, and we collected some animals or bacteriums under this consent or contract, and then it's recorded. So that's the important point. Okay. So, but anyway, I think probably recently Indonesian science is getting stronger. So maybe, we don't, I think the Indonesian scientists should use your own resources by yourself. So that's the point. Okay. Anyway, this ABS was adopted in 2010 and entered into force in 2014. So, well, it's, some people says, due to the ABS, people are reluctant to go to the foreign countries to collect animals. Yeah, that is true. But I think more collaborative work is needed to do the research under the ABS. Okay, so then how about the current status of Durastatin 10 related drugs? So actually recently, Merck, big pharmaceutical companies, announced 4.3 billion investment to Seattle Genetics. Okay, so this is really ironic, okay, because all of big pharmaceutical companies abandoned natural products research. Okay, so then, yeah, Merck did too. Merck had a very good natural products research group, which led by a person who had who shared the patent of Durastatin 10. Okay, then, but of course, he left because Merck closed the natural product research group. Then now they paid $4.3 billion to the small companies, okay, which still continuing the natural product research. Okay. So now, especially in the United States, many of small companies are doing natural product research. Then when they found they developed a good drugs, big pharma buy it. That's the current situation of natural products development. Okay. Also, around 30 drugs, mostly antibody drug conjugates of durastatin 10 are in clinical trials. So more drugs will come. But also, right now, ADC research is mainly about durastatin 10. Okay, so durastatin 10 is a very good example for ADC, but also probably we need to develop the other compounds by using ADC technology. Recently, not last year, 2 years ago, other drug, Polivy, was approved. So this compound is also consists of MMAE and monoclonal antibody which targets CD76B. Okay? So then CD63B, this antigen is expressed in the other type of cancer cells. So that's why this drug treats non-Hodgkin lymphoma, chronic lymphocytic leukemia, and B-cell lymphoma, which is different from adocetris. So adocetris can treat Hodgkin lymphoma and anaplastic large cell lymphoma. Okay, so the target cancer cells is different because target antigen is different. Okay, so probably more different drugs which target different cancer cells will be marketed soon. So I think I need to finish maybe within 20 minutes. Well, 25 minutes. So let me, where am I now? Almost time. I'm gonna finish soon. Okay, so another recent example is anti-cancer drug derived from colonial acidium. So this colonial acidium, or tunicate. This tunicate name is Ectanacidia turbinata. Okay. So ectanacidin 743, or we can call it ET743, or another name, commercial name is Yondelis. So this compound was very potent. Then this compound is now approved as an anti-cancer drug of soft tissue sarcoma and ovarian cancer. And then this compound is alkylating drug that binds guanine residues in the minor group of DNA. So the biochemical target is a groove of DNA. So that's why it can inhibit the DNA synthesis. Okay, so then let me check. So this is the history of Yondelis, or another name is trabectedin. Okay, actually, it was approved 6 years ago, but when the activity was found in this tunicate, was 1969. Okay, so it means 46 years is needed after the screening. Okay, but actually structure elucidation, even structure elucidation was very difficult. It took 15 years. Okay, so this is very good structure elucidation in 1984. After that, I think Dr. Ken Reinhardt determined the structure. The Dr. Reinhardt had worked at University of Illinois. So University of Illinois sold the license to Pharmamar, which is a Spanish company. The company focusing on marine natural products in Spain. Okay. Then in 1994, then they asked the very famous organic synthesis chemist Dr. E.J. Corey to synthesize this compound. So total synthesis was achieved in 1996. But no, the total synthesis, although the halogen was manufactured by the total synthesis, Pharma cannot use this total synthesis method. And also, 2011, the production of symbiont was reported. So here, they isolated this compound from this tunicate, but true producer is not a tunicate, but a symbiotic marine organisms. Its name is Candidatus endoectenocidea frumentensis. So Candidatus means there is a no type strain because we cannot culture these microorganisms. So we can pick up or isolate the cells of these organisms from the tunicate, but we cannot culture them. Okay, that's a very, very difficult. There are actually many of sponge metabolites are produced by the symbiotic marine organisms, also tunicate as well. But it's really difficult to culture them. So if you remember the helicondrin bee, that is also cultured, probably produced by the symbiotic microorganisms. But still, we cannot know which microorganisms produce the helicondrin bee because it's difficult to symbiotic microorganisms in the sponges. Okay. So anyway, production by symbiont was reported, but they cannot produce by using the fermentation technology. Okay. Then instead, after the total synthesis, they found this technique. They developed this technique. Cyanosulfuracin B, which was produced by Pseudomonas fluorescens. So we can culture them. Then by doing the fermentation of these microorganisms, we can produce cyanosulfuracin B very easily by fermentation. Okay. Then they are doing semisynthesis. They can modify these compounds into externizidine 743. So it's much easier than total synthesis. So right now, Pharma synthesize these compounds by semi-synthesis of fermented chemicals. Then it was approved in 2015. Okay. So symbiosis is, is a very good, important keyword in marine natural product chemistry. So, of course, without culturing the symbiotic organisms, we can extract the DNA, right? We can extract the DNA of synthetic organisms. So that's why biosynthetic work can be achieved by using the genetic data. Maybe in the case of ET743, biosynthetic has not been proved by experimentally, but some of symbiotic productive compounds was investigated for its biosynthesis. Okay, already. So let's see. Again, I think in the introduction, I explained that some of the data came from the website of marine pharmacology. So the website exists in the Midwestern University in the United States, but you can just Google it by the keyword marine pharmacology, and then you can find this website very easily. So there is some information to general public, or you can find the clinical pipeline list or preclinical pipeline. Yeah, I think it's good to go to the clinical pipeline to find these compounds. Okay. So also, if you go to the seminars, you can watch the YouTube of the lectures. Okay. So then, so right now, I explained the approved drugs, right? But many compounds are now under the clinical trial. So you might know what is a clinical trial, but just briefly, there are some phases, maybe phase 0 or sometimes phase 4, but mainly phase 1, 2, 3, right? Phase 3, they do the screening for safety. In the phase 2, they assess the dosing requirements and they determine the efficacy. So in the phase 3 is the final confirmation of safety and efficacy. And they test with large group of people. Yeah, so maybe 30, 1,000 people. Sometimes, 10, more than 10,000. Okay. So actually, yeah. Then this is a number of clinical trials of marine-derived drugs. So, 4 drugs are under the phase 3, 12 drugs are under the phase 2, 7 drugs are under the phase 1. But actually, the clinical trials, this is a number of drugs. So clinical trials have been done in different countries by different companies. So the number of clinical trials is maybe 100 or more. Okay. So I want to just show you one example from the compounds under the clinical trials. So cyanosporamide A, okay, this compound, okay, and commercial name is marizomib, okay. This compound was isolated from Salinispora tropica in 2003, very recent, okay. Maybe 18 years ago, so maybe it's not recent, but for us, it's recent, okay. Yeah, actually, many of currently approved drugs were reported in 70s or 80s. So compared to these compounds, it's very recent compound. Professor Bill Fenical in Scripps Institution of Oceanography and University of California, San Diego, he reported the structure of this compound. Sarnispora tropica is a marine actinomycete. So he collaborated with Dr. Paul Jensen in the same institute. So the Paul Jensen is a microorganism the scientist. Yes. So then that time, maybe, yeah. So then this was very potent. Actually, the structure is similar to omuralide, which is a proteasome inhibitors. So this compound actually inhibit the 20S proteasome. Okay. Then right now it's under the phase 3. Then target cancer is non-small cell lung cancer and pancreatic cancer. Okay. So pancreatic cancer is very difficult to treat. So if it can be developed, it's very good hope. It can be a very good hope. Okay. So in this case, actually, Salinispora tropica is, I think, was isolated from the sediment. Okay. So not a symbiotic bacterium. Okay. That's why it's culturable. So we can culture them. So then we can produce this. I think the company produced this compound by fermentation. Okay. So, yep. So then if this compound is approved, this would be the first marine microbial natural compound to be approved. Okay, so the other compounds are derived from microalgae or animals, not microorganisms. But this is a very good example of potential of marine microbials. Okay. Again, you can see the chlorination. Okay. Actually, biosynthetic work was done by Dr. Brad Moore in the same institute. Okay. So, like I said, this actinomycete is culturable, so they can do the biosynthetic work very easily. Okay. Yeah. So, okay, I think this is the final slide. So maybe I wanna explain the future of potential of marine products. Okay, like I said, when I was a student, 3,000 marine natural product was reported. Right now, 36,000 of marine natural compounds have been reported. Okay, then 15 to 20 compounds have been approved as drugs. Okay, so I'm not sure if the number is few or not, but I'm sure it's growing. Okay, so recently, in the last 5 years, if you go to the marine pharmacology website, recently more compounds are approved. So you can realize it's increasing recently in the last 5 years. Okay. According to the census of marine life, 250,000 species of marine organisms have been reported. This number doesn't include microorganisms. Okay, so then these sensors predict 760,000 species have not been reported. So it means 1 million species marine organisms exist. Okay, 1 million of macroorganisms. Then presumably more than 1 billion microorganisms exist in the sea. That's the prediction of Census of Marine Life in 2010. Okay. So considering this situation, we can predict. Actually, I forgot who mentioned this one, but marine organisms include 150 to 600,000 new compounds. Currently 36, right? Maybe 10 times more compounds will be reported or can be reported. So now we have more potential. So also we can use ADC technology. So I wanna continue to look for the new compounds from cyanobacteria, and the other people will continue it. Then I hope we can, marine natural products can save more people, the life of more peoples. Okay, thank you. So I'm very happy to answer your questions.
Okay. Okay, thank you very much. Sorry. So because I have to work from home. Wait a minute.
Okay, for all the audiences, that's from Professor Tatsufumi Okino, and it is time for the question and answer session. And I would like to read some of the questions.
Bapaknya kenapa ya?
Okay, I would read question from the chat in the Zoom. The first one is from Anissa Bella. Hello, I am Bella. Before I ask a question, thank you for Professor Tatsufumi Okino because the lecture material presented is very interesting. So I am curious about antibody drug conjugates, as we know that ADCs represent a new class of cancer therapeutics. The design involves a tumor-specific antibody, a linker and cytotoxic payload, and also designed to allow specific targeting of highly potent cytotoxic agents to tumor cells while sparing normal cells. I want to ask, is there any method to reduce the toxicity that occurs in order to increase ADC therapeutic index in the future? Thank you. For the professor, maybe if you would like to read the question, it is on the chat from Anisabella. she asked about, is there any method to reduce toxicity?
Yeah,
in
thank you for the question. Actually, we don't have to reduce the toxicity because the antibody drug conjugate is carried to the cancer cells. So in the cancer cell environment, it should be toxic to kill the cancer cells, right? So if toxic to normal cells, it's not so good, right? But the antibody can carry the drug, and then drug is released is only in cancer cells. So we don't have to reduce the toxicity. Or antibody, drug delivery by using antibody is a mechanisms of reducing the toxicity. Yeah.
Okay, thank you, professors. And then the next question is from Muhammad Sunimawan. So in Indonesia, there is a multivitamin from Japan called SOP 100 Plus containing salmon AFC, which is consumed by many people. It is said to have a good effect in battling COVID-19. So, what do you know about this vitamin and what do you think about it? Thank you, Professor.
Well, um, so I'm it, to be honest, I don't know that information. So in Japan, such a vitamin is we don't have any information that such vitamin is good for the COVID-19. So I can say if it's really, really effective, probably we can use it, right? Well, it should be more popular in our country too. So, but basically, you know, to fight to the COVID-19, our immune system should be very strong, or we should be very healthy, basically, right? So if we get a small amount of the virus and then we are healthy, we are very good. If we have a good immune system, we can fight. But maybe some vitamins or some such a you know
Okay,
chemicals
so
can help to keep the healthy, but but I don't know the direct effect to the COVID-19.
okay, thank you, Professor. And for the next question, maybe it will be led by Mrs. Anissa to encourage students to ask directly.
Okay. Yes, thank you very much, Professor Okino. But maybe Sensei, we cannot see. Maybe can you turn on your camera? I think we cannot see you.
Really?
Yes.
I thought. Hmm, I can see my
Oh,
face.
okay, maybe. Yeah.
Hmm,
Okay, I think
actually,
that's why.
I cannot control it.
Okay.
I don't know.
Yes. Okay, that's
Just a
fine, Sensei.
moment. Hmm. Can you hear me?
Yes, we can hear you. Your voice.
It says no response.
Oh, that's fine, I think. Maybe Mr. Wanda, how about in your monitor?
Actually, professor, I think there is something blocking your camera because
I see.
in the Zoom, your camera is on
But just dark.
and just that. Maybe something blocking your camera.
Yeah, something is strange actually.
Okay.
Because I cannot control anything.
Or maybe
So
turn off the camera and then turn on again.
No, I cannot. I cannot turn off the camera.
Okay. Okay, that's fine, Sensei. Yes, so this is a question from Ahmad Zaki. Zaki asked about this one. If you want to test a substance from a species, let's say from Cnidaria, how do you know what the pharmacological effect it does, like antiviral, for example? Or we can just randomly test or match it with non-existing substances. Yeah, I think he wants to know how to do the bioactive research, maybe. Okay, Professor Okino.
Okay, just a moment. But anyway, it so my Zoom system is recovering. So, but sorry for not showing my face. But actually, it's really difficult questions. So personally, no, I want to avoid that what the other people did or what the other people are doing. So if we do the same assay system, if we test the same biochemical test, we can get just the same result. We repeat the same result, right? So that's why if we have a unique biological testing, it's very good. But if we can use a different source, I think you might be find a different result. So that's why it's or just it depends on your philosophy. For example, if you, in my opinion, I'm thinking to start the antiviral drug research because of the current situation of the world. So, but of course, I cannot use the virus itself, but I will, maybe I might use some biochemical target. So, or, if you have some philosophy, you can find the target by yourself.
Okay, so maybe it's better to find from different source, and then we can make an experiment, and we hope that it shows a different result. Is it?
Yeah. Yes.
Okay. Yeah, that's the answers, Zaki. Okay, I think it's clear. And the next questions from Ibu Sri Kadarinah ya. Udah nggak? Ya. Okay. This is a questions from our lecturer, Ibu Sri Kadarina, and previously she was a pharmacist in oncological in hospital in Yogyakarta. So in public hospital in Yogyakarta, and she's one of the pharmacists in the oncology. So I think she also interested about the anti-cancer from the marine product. So the question is how to get large material and how to make the medication like that. Also, I think the time, what time it takes to do the experiment. I think from the collecting until we can get the drugs, and then maybe there should be make cooperation with the government. So, what do you think, Professor Okino?
Well, well, I think the question covers many different topics. So, well, but actually, if you remember my lecture very carefully, the old compounds took longer time to develop. Then recently, for example, salinosporamide, the research is very quick. So right now, probably 30 years ago, it was really, really difficult to do a research. But now, we have lots of technologies. So, but also clinical trials, it's really, really expensive. So if you look at the list of clinical trials, many companies are waiting to get investment, right? So in the United States, they have a very good success. So because not government, of course, the government invests a lot to National Cancer Institute. Then, yeah, actually, Dorastatin 10 or Halichondrin B or yeah, many compounds were supported by National Cancer Institute. Yes, but also many people or many companies invested to the small companies which are interested in the interested in the marine natural products. So yeah, so investment to small company is also important. So yeah, it's like, and then currently, large scale collection or sampling is really difficult. So probably, I think we should abandon that kind of idea. But if most of compounds are produced by microorganisms, so we need to find the method to culture symbiotic marine organisms. But it might be possible. For example, dinoflagellate. Dinoflagellate are famous, one of phytoplankton, but some of dinoflagellate are toxic. So it's it's famous for the marine toxins, right? Then some of dinoflagellate are difficult to do the arsenic culture. Okay, so without existence of microorganisms, it's difficult to culture the dinoflagellate. Why? Because then recently some scientists found that microorganisms supply the nutrient, right? Then actually if you they investigated the genome of dinoflagellate, and then they found that these dinoflagellates lacks the genes to synthesize the nutrient. Then instead of they synthesize the nutrient by themselves, they get the nutrient from the microorganisms. So in this case, if you provide the nutrient as a chemicals, you can culture it, right? So that's why it's the culturing of symbiotic microorganisms not impossible. It's possible if you provide the proper culture conditions. So probably if we develop the culture method of symbiotic microorganisms, we can explore more natural products from marine resources.
Okay, so the key is culturing the natural products, and I think how to get the proper nutrition is very important, right, Sensei? Because I think if the environment is not the same as the nature, so I think the substance also will be different. But what do you think about that?
Say it again.
So I think the proper nutrition for the culturing is very important, isn't it?
Yeah, yes.
Yeah. So how to how to how to how to make sure that the the proper nutrition is the proper nutrition for the substance for the marine products, or we can just uh, mix and just how to say imitate like the natural in ocean, or how to do the culturing.
Understand. Well, of course, it actually is, the different case have a different conditions. So it's, we don't have a general rules. So that's why it's difficult, right?
Yes. Okay. So yeah, that's the answer, Ibu Sri Kadarina. So so to make a large to get the large material. So now the researchers make a culturing for the symbiotic with the proper nutrient for the growth of the natural product or marine natural product. Okay, so we'll go the next. Is there any question? Okay, maybe from Aini. Yeah. When you decide to derive some substances to another new substances, did you just randomly test it? I think it's almost the same with Zaki's questions. Is it any? Yeah. Okay, maybe Sensei, do you want to add the answer from Aini's question?
Repeat the question.
So if when we decide to derive some substances to another new substances, did you just randomly test it, or there are any specific process? Maybe she wants to know about the from origin substance and how to make another new substance. I think this is synthesis. Okay.
But anyway, the I'm not sure I can answer that correctly, but it's difficult to make a decision. For example, we can isolate very good compounds, right? Then to develop the compounds, we need to choose a compound. Then we need to invest a lot, right? If we start invest the compound, developing in a clinical trials or so, it takes a time, right? Maybe we might stop the screening experiment, right? But that, if it's failed, we need to find a new compound, right? So, well, it's really difficult for us to stay at a screening level or to go to the clinical trial levels, right? So of course there are more multiple levels. So probably, especially development of drugs needs lots of money. So let's say if we devote ourselves to develop, we need to devote ourselves to develop the drugs. Without such a attitude, we cannot develop the compounds to the drugs. So it's really, without making a big decisions, we cannot develop the compounds. So it's really, when to make decision is a very big problem.
Okay, thank you very much, Sensei. Yeah, I need. So there are so many level, and we need to before we decided before we decide to do the next level, we have to think many times, and we have to confirm many things before we go to the next step. And maybe this is the last. Is it okay, Mas Wanda or Mbak Devi, or maybe this is the last? I think we have the last question. Is it okay? Okay. So this is from Haider. I want to ask, in the current condition, corona pandemic, is there any marine biotic material that could potentially be an effective anti-coronavirus drug? If so, what is the potential for the development of anti-coronavirus? Okay, Professor Okino.
Yeah, actually, some of the compounds have a potential. Like I said, catepsin inhibitors. We have cyanobacterial compounds which inhibits the catepsin inhibitors, catepsin L inhibitors. So catepsin L can be a target of anti-COVID drug, but well, I think to develop new compounds, it takes at least 5 years. So I hope 5 years later, we don't need anti-COVID-19 drugs. So, but there are some example, but it's a early developmental stage, so it takes time.
Yeah, it takes time, and it's still possible to get the anti-coronavirus medicine from marine product. And as Sensei mentioned before, that from California, also the researchers found that some natural products can inhibit the COVID-19, but still, because COVID-19 is still new virus, and so many research still ongoing. So yeah, I think it's challenging, and we hope that coronavirus will disappear from this world. Okay, thank you very much, Professor Okino. Thank you very much for the interesting lecture today, and thank you for the students, lecturers, and we hope this lecture can widen our knowledge about natural products. I think this is the end of the session, and we will take a photo together for documentation. Can you turn on your camera for the students, please? Mr. Wanda and Ms. Devi will take our photo.
Okay, everyone, ready? Okay, smile. 1, 2, 3. Okay, one more. One more. Okay, 1, 2, 3. Smile. Okay, thank you.
Okay, finished. Okay. So, Professor Tatsufumi Okino, thank you very much for your time and for your great explanation about opportunity of the marine natural products in the pharmaceutical science. Me as
You're
the,
very welcome.
yeah, okay. Do you want to say something to close?
Well, can you see my face?
Still like dark.
Oh, really?
Yes.
I'm very sorry about that. So
Okay.
yeah, well anyway, if I have a chance, I wanna visit UMY in, I don't know, not this year probably,
Yes.
but in a few years basis. So yeah, I'm very appreciate that maybe more than 200 students attended. So it's really, I'm very glad to give a talk to such a large audience. Yeah, thank you so much.
Thank you so much, Professor.
Yeah, this is Mrs. Pinasti Utami, the coordinator of this event. Can you turn on the camera? Can you turn on the camera, Mrs. Pinasti? No?
Kamera rusak ya, Mbak.
Okay. Okay.
Arigato gozaimashita.
Arigato gozaimashita. Okay, we also want to go Hokkaido, Sensei.
Yeah,
And thank Hokkaido University and Sapporo and also Japan, because yeah, we know that Japan is really nice country. Okay, I think this is the end of the session. Thank you very much for your attention. We close this class with saying Alhamdulillah together. Alhamdulillah.
Alhamdulillahirabbil alamin.
Yeah. Okay. Thank you, Professor Okino. Hope you can get healthy and please take care. Goodbye, Sensei.
Bye. Thank you. Bye.
Selesai guru ya.
Issued by the Director General of Higher Education from the Ministry of National Education, Indonesia, dated on 9th of July 2010, has become its legal basis for the establishment of the School of Pharmacy from the Faculty of Medicine and Health Sciences at University Muhammadiyah Yogyakarta. It was followed by the establishment of the School of Pharmacists on November 2016.
Our curriculum is competency-based curriculum with program-based learning method in which study program of Universitas Muhammadiyah Yogyakarta is a pioneer implementing the method in learning pharmacy in Indonesia. Pharmacy study program and pharmacists have several adequate laboratories to support learning process and have partners for students practicum such as hospital, primary health care centers, pharmaceutical industry, and pharmacies.
School of Pharmacy is one of the newly established study programs in Faculty of Medicine and Health Sciences at University Muhammadiyah Yogyakarta. The decree issued by the Director General of Higher Education from the Ministry of National Education, Indonesia, dated on 9th of July 2010, has become its legal basis for the establishment of the School of Pharmacy from the Faculty of Medicine and Health Sciences at University Muhammadiyah Yogyakarta. It was followed by the establishment of the School of Pharmacists on November 2016.
Yes, lecture untuk hari ini sudah selesai. Terima kasih untuk para mahasiswa, Bapak Ibu dosen yang telah hadir. Terima kasih atas waktunya dan kita akan akhiri Zoom-nya.